Structure of human sperm. Let us discuss about the structure of human sperm. So it is a continuation of my last class. So human sperm is a microscopic structure, elongated and microscopic structure. The entire human sperm is covered by plasma membrane. It is covered by plasma membrane, and it is structurally divided into head, neck, middle piece, and tail. Head is the anterior part, short neck which connects middle piece and head, and middle piece is the middle portion, and tail is the posterior or last portion. The anterior portion of the sperm. So this is the structure of sperm, human sperm. The entire sperm is externally bounded by thin plasma membrane, and structurally it is divided into head region. So this one, and short neck which connects head and the middle piece, and middle piece, and this is elongated tail. The anterior region, head, is elongated and it encloses a haploid nucleus, single haploid nucleus, and this haploid nucleus is elongated in structure, in shape it is elongated. It contains 23 chromosomes. Then anterior portion of this head has a cap-like structure known as acrosome. It is known as acrosome. So this acrosome encloses the enzymes. It encloses the enzyme necessary for the fertilization. It is necessary for the fertilization. During fertilization, the sperm gets attached to the outer membrane of the ovum. Okay, so this attachment and penetration of the sperm into the ovum is due to the activities of enzyme present in the acrosome. Okay, next region is the neck, which contains centrioles, small portion which connects head and middle piece. Then the middle piece has many number of mitochondria. It has many mitochondria. It is rich with mitochondria which are spirally arranged. And this mitochondria is the source of energy. It is essential for providing the energy necessary for the motility of the sperm, necessary for the activation of the sperm during fertilization. The last portion is the tail, which is elongated. Elongated tail is there. So this tail is also helpful in the movement. It helps in motility of the sperm. So this is about the structure of sperm. The sperm released by the testis, seminiferous tubule, are added with the fluid secreted by the accessory ducts and accessory glands. Mainly the seminal vesicles, prostate gland, and vas deferens and epididymis secretes enzymes and other materials, and these materials are generally known as seminal plasma. The sperm released by the seminiferous tubules will be added with the seminal plasma, and now it is called as semen. It is known as semen. So semen is nothing but the secretions of accessory glands and accessory ducts along with the sperm. Is the semen. A normal human can ejaculate about 200 to 300 millions of sperm during a coitus. Out of this 200 to 300 millions of sperm, 60% is very essential for the fertilization, because at least 60% of the semen should be active semen, and this condition is very essential for the fertilization. So this is about the structure of human sperm. So during last class and this class we have learned the gametogenesis in man, the process of formation of gamete in human being or human man is known as spermatogenesis. We have learned the process of spermatogenesis and structure of sperm. Let me explain about the oogenesis. Oogenesis can be defined as the process of formation of ovum. The oogenesis occurs in the ovaries. As we know, paired organs. Ovaries are the paired primary sex organs in human female. Okay. The oogenesis, there are differences between spermatogenesis and oogenesis. The pattern of spermatogenesis is different from the pattern of oogenesis. The oogenesis begins from the fetal stage, or during the fetal development, whereas spermatogenesis starts when the male attains puberty. The spermatogenesis starts. However, oogenesis begins at the fetal developmental stage. Okay. So during the fetal stage of development, couple of millions of oogonia or oogonial mother cells will be formed within each fetal ovary. Then these cells starts division. So oogonia. So here it is that. So these oogonial cells starts division during the fetal developmental stage, and when it reaches the first stage of the meiotic division, it gets temporarily arrested. So starts in fetal stage, number of oogonia or oogonial mother cells will be formed. Millions of oogonial cells will be formed, and it gets arrested in the first meiotic division. It gets arrested during the first meiotic division. So no more oogonia will be formed after the birth. After the birth. Okay. Then, so this oogonia later on starts division and enters the prophase first of the mitotic division and gets temporarily arrested at this stage. And these cells are known as primary oocytes. Then it is known as primary oocytes or primary oocytes. Primary oocytes. Okay, and later on, this primary oocytes after the birth of female child. So these primary oocytes are surrounded by single layer of granulosa cells. They are surrounded by the single layer of granulosa cells. Now it is said to be primary follicle. Primary follicle. So primary follicle means it is a primary oocyte surrounded by single layer of granulosa cell. So the primary follicles will be formed after the birth. Okay. Then a large number of primary follicles degenerate during the phase from birth to the puberty. A large number of such primary follicles will be formed in the ovary. However, all these primary follicles cannot mature and release the ovum. Most of these primary follicles, a large number of primary follicles, will be degenerated from birth to the puberty. Okay. At puberty, only 60,000 to 80,000. At puberty, only 60,000 to 80,000 primary follicles will be left in each ovary. Then the primary follicles get surrounded by the more and more layer of granulosa cells, and a new theca is formed. Then it is said to be secondary follicles. Secondary follicles are nothing but the primary follicles, which is covered by many layer of the granulosa cells, and a new theca is formed. Then the secondary follicle soon transformed into a tertiary follicle, which is characterized by a fluid-filled cavity called as antrum. So here, so this is the oocyte. Okay, this is the oocyte, primary oocyte. It is surrounded by the granulosa cells. So these are the layers of granulosa cells. Different layers are there, and there is a cavity will be formed at the center. Okay, and this cavity is known as antrum. Is known as antrum, and this antrum is filled with a fluid known as antral fluid. Antral fluid. Okay, so this follicle is known as tertiary follicle. Tertiary follicle. Tertiary follicle. Okay, tertiary follicle will be formed. Then later on, this tertiary follicle will be undergo further changes, further development, and it will be converted into mature follicle or Graafian follicle. Graafian follicle. During the formation of Graafian follicle, some other changes occurs in the tertiary follicles. Mainly, another layer of cells will be formed around the secondary oocyte of the secondary follicle. Oocyte of the secondary follicle. Okay, it is the secondary oocyte, and this layer is known as zona pellucida. Zona pellucida will be formed, and once again around the granulosa cells also the thecal layer gets differentiated into outer theca externa and internal theca interna. Outer theca externa and inner theca interna. So these are also formed. Okay, so such follicle is said to be Graafian follicle or mature follicle. Fully matured follicle. Okay, then the oocyte which is present inside the Graafian follicle is said to be secondary oocyte. Secondary oocytes. During the formation of the secondary oocytes, so what happens? The primary oocytes undergo meiotic division. First meiotic division. Once again, first meiotic division, and it forms a polar body. It forms a polar body and secondary oocytes. Secondary oocytes. Okay. Then, so these secondary oocytes next undergo second meiotic division. Second meiotic division, and it results in the formation of second polar body, and mature ovum. Mature ovum. Ovum will be formed. Okay, so this mature ovum is present in the Graafian follicle. Then, so this Graafian follicle ruptures and releases the ovum. Later on, the Graafian follicles ruptures and it releases the ovum, and this process is known as ovulation. It is known as ovulation. Ovulation means the process of release of ovum. The process of release of ovum from the Graafian follicle is known as ovulation. So this ovulation occurs from the ovary. Okay, after the ovulation, the remaining portions of the Graafian follicle, that is the granulosa cells, theca externa and theca internal cells. So these structures undergo differentiation. They undergo differentiation to form a glandular structure known as corpus luteum. Corpus corpus luteum. Corpus luteum. So this corpus luteum secretes progesterone. It secretes progesterone hormone. Progesterone, a steroid hormone. Okay, so this is about the process of oogenesis. Process of oogenesis.